All right, so this week on Monday and Friday we will have two sort of seminars, but this is meant to be seminars where you sit there with the paper in front of you. Now I realize you've just gotten this paper, so you haven't had a chance to look it over yet. But maybe you've had a chance to look at the figures and stuff like that. That's what I want you to do, actually, as we go through. So feel free to take notes as you go through. This is how to read a paper, and we're going to have two papers. This one's on the structural end of things. The next paper is more on the systems end of things. It's more about a bunch of different things that you're not looking as closely at. This is one that you're looking closely at one thing. But they are similar because they are done by the same person, James A. Wells. And I just saw him actually at the conference that I was at New Orleans. And I thought about how I found out about the new work he's doing, and so I'm going to introduce that to you on Friday. But the reason why I want to do that is because I've taken this paper as a great example of the old work that he did. And there's another couple of connections that we have with our class. So I'm going to show you some sections from the paper. I'm going to sort of translate. We're going to have a lot of text on some of the slides. The point is not to read the text as we go through, but for you to sort of use that as a way to read the paper eventually. I gave you it on paper, so you can actually write on the paper if you want. That's what I like to do when I read a paper. You do it however you want, though. Now, in general, this paper is a little bit older than what most people should be using for their protein design presentation because it's almost 20 years old. There's a lot of protein design that's happened since then, and so you probably will be going for something a little bit newer. But I'm doing this as a sort of foundational knowledge about protein design. Many of the concepts of analyzing how a structure works, how a design worked, will be common. The other thing about this design uses random variation to design a binding peptide. So it's not rational. I mean, I wouldn't turn you away if you had chosen this paper and you wanted to do it. I wouldn't turn you away because I'm not interested in saying, oh, that's not rational at all. There are some rationalities to their design. You basically need a strategy to talk about. If they don't have a strategy, if they're just trying random stuff and seeing what works, that's not much of a strategy. So you'll have more to talk about the more rational the design is. Even in this one, however, there's enough rationality to talk about that you can really talk about what their strategy was. And I'm going to show you what that is. But what this is, the reason why I like this paper so much is it's sort of a counterfactual game. Historians actually don't like to admit that they play the counterfactual game. It's a little bit embarrassing to them. But still, it's the same kind of thing, and you see it all over in literature and pop culture and things like that. What could have changed history? What could have changed what happened? Did things have to happen the way they did? Or could anything have just happened? What are the boundaries to reality? Okay? And in a sense, if you've read enough of the book, you may have gotten into the point that this is one of the underlying themes of A World from Dust as well. But we're going to start off with what is an experiment that asks this kind of question. Now, historians get to have fun with this. And there's a bunch of historians who wrote what if, military historians particularly, because a battle is something with an undetermined outcome that can be changed by just a few things changing. So the military historians love to focus on a particular battle and say, well, what if the other side had won that battle? What would have happened to the world? Now, there's counterfactuals and there's counterfactuals. Sometimes they say the battle would have changed, but the war would not have changed. In fact, there's a question about that. So here's some examples of chapters from the What If book. Infectious Alternatives, the plague that saved Jerusalem in 701 BC. This is about not when the Babylonians attacked Israel, that was later, but this is earlier when the Assyrians attacked Israel, and technically the Babylonians attacked Judah because when the Assyrians attacked, they attacked from the north, and they basically destroyed all the northern tribes of Israel. The 10 tribes of Israel were not really around anymore after the Assyrians attacked. And they attacked all the way down to Jerusalem. They besieged Jerusalem. The story is, I believe, in 2 Kings, where they're running out of food and people are like having to eat donkeys and stuff like that, which is not exactly in the middle of the Hebrew religion, right? So they're talking about this historical event, and what the Bible says is it says that the angel of the Lord slew a large number of the Assyrian army overnight. And when they woke up, they thought they were still besieged, And then there's a couple of lepers, I think it's 4 lepers, that decided to go out because they're like, okay, we're going to die here anyway, so we might as well just go over and turn ourselves over to the enemy. And they go over there and everyone's dead, so they like start feasting, and then they're like, oh, we really should go back and tell everyone else. It's a very dramatic story. But you have an army that is being destroyed by an external agent in a very short time. So people look at that and they say, well, it looks like a plague, and I have no objection to that. The Bible says that God worked through that event, and he could have worked through a plague as well as he could have just snapped his fingers or whatever. The real point of this is not diagnosing what plague it was. The real point of this is diagnosing its effect on history. What would have happened if Jerusalem had been destroyed by the Assyrians? Which, as you know, if the 10 lost tribes of Israel were lost, what if it was 12 lost tribes of Israel? Because the Assyrians destroyed the peoples that they conquered. Babylonians, on the other hand, had a sort of weird exile and preservation strategy. Because the Babylonians were the ones who conquered Jerusalem a number of decades later, then the Jewish culture was preserved. What would the world look like if Jerusalem was destroyed at this point? Alexander the Great, what if he died before he conquered India? He died young as it was, and the event was like a stomach ailment or something like that. But he got all the way out to India, and he basically united. It's why the New Testament is written in Greek, because he conquered Palestine. So what would have happened if he had died before, when he had only conquered half the world rather than the whole world? That kind of thing. What if Napoleon won at Waterloo? Could have happened. Whenever you have a battle, you could have had a brilliant strategy, especially a brilliant strategist like Napoleon. There's this one. If the lost order hadn't been lost, then Robert E. Lee could have humbled the Union in 1862. That would have been, this argues that the Civil War would have had a different ending. And that obviously has huge repercussions. This is one that is a little more recent. A couple of movies about this era that were up for Oscars last year. What a Taxi Driver Hath Wrought. I believe it's 1938 or so. It's the late 30s. But it's not 1939, because in 1939, Winston Churchill was in England, and he was clearly the leader of the English when the Germans were threatening in World War II. Before that, a few years before that, he was visiting New York City, and a taxi cab hit him. It didn't really injure him that bad, but if it had been another inch over, it's the kind of thing where frequently people would have died from that kind of impact. What if Winston Churchill was not around in 1939 to go through the events of Darkest Hour, or to provide the leadership that said, for Dunkirk, let's evacuate using civilian boats, stuff like that. Lots of history here. But the thing I want to point out is about Lee's Lost Order. I just want to go into this in a little more detail. This is Lee's Lost Order was in the Civil War, 1862, and the Robert E. Lee had a triumphant Second Manassas campaign. He was laying plans to win the war. Now, the North always had all the advantages, but they had bad leadership early on. And so they lost a lot of battles. I really think, personally, I think it really was bad leadership from the North, not good leadership from the South. People argue about that all the time, and I can lay out my argument another time. But basically, for whatever reason, the South, even though it shouldn't have threatened the North, was actually concentrated in Maryland. Okay, so it's in the North, 40 miles northwest of Washington, D.C. And the whole thing was, if they could besiege Washington, D.C., that's the whole thing. If you look at the war, Washington, D.C. is right on the line between North and South. So it's a clear military objective for the South to besiege the North's capital, okay? The national capital that they did not recognize. So actually, and of course the Union tried to take Richmond, so the South is trying to be a victory away. He was actually approaching from the northern side, he was approaching, and you had the Battle of Antietam, which was like Gettysburg a year later. It's one of the great turning points of the war. Now, lots of turning points in a war, but the Union was demoralized. Lee was outnumbered, but he might have a realistic chance of winning because, after all, he had won in previous circumstances. And he was on the move, and they didn't know where he would attack from. But there were events that happened that allowed the Union to eke out a critical victory. It was one of those victories that was so costly that it was kind of like, is this really a victory at all? But it ended up being a victory because Washington, D.C. was not besieged. It centered around 2 sheets of paper. Early in the morning of the 13th, Corporal Barton Mitchell, the 27th Indiana, he was looking outside a recently vacated Confederate camp, and he found a bulky envelope, paper wrapped around 3 cigars. The document was by the command of Lee, signed by the Assistant Adjutant General. It was passed up, and they basically verified the signature. It was an authentic copy of Special Order 191. Special Order 191 told the rebels, this is the way we're going to attack. So the Union was able to know where they were going to attack. It headed them off. The Union was able to make a battleground of its own choosing, and the Union, even with all these advantages, the Union barely won. But they did win, and they repelled the South's attack, and eventually the North won the war. Would this have been enough to change history? Well, the North still had so much advantage and so many soldiers and so much more factories and things like that, that you could argue either way. But if you talk about something where, what if he had walked right past that bulky piece of trash, it looked like? Things could have been very different, big consequences. And of course, the Star Trek reboot, now they're rebooting everything, but with Star Trek they actually have time travel, so they can literally reboot, and they go back and they say, well, what if Kirk and Spock went through the events of their 5-year mission differently? Of course, they squandered it entirely with the second movie, but that's another story as well. So you can be grumpy about this. I kind of like the third one, though. But that's really not what we're here to talk about. We're here to talk about what can we do. We can't wind back time with a protein, but we can re-evolve it. So maybe instead of running history again, we can run natural history again, and we can say what's going to happen with a protein. So the protein that we're going to focus on is the Fc region of an antibody. Now, we're going to talk more about structural immunology and what antibodies look like in a second, but there's some rudiments of antibody structure that we're going to have to talk about today. All antibodies have a grasping end that's supposed to bind something, and all antibodies have a non-grasping end that's kind of like their handle. If antibodies are supposed to grab onto stuff, how do you grab onto the antibody? Well, they have different fingers, different binding sites, so they can bind different things. But they're all the same on the back end, at least within a particular subtype of antibody. That's why they call it the constant region. And so the constant region, where all antibodies are the same. So the thing is, we see a bunch of proteins that grab onto this handle. Now, do they grab onto this handle just because they've been told to grab onto the antibody there? Or is there something about the handle that is especially grabbable? You see what the question is? Are they forced to grab the handle there and there's just nothing special about it? Or is there something special, like something sticky or binding properties that make that handle the best place to bind the protein? And so the question that they asked is, what if we made up a random set of peptides, and the only rule was that they had to bind the Fc region somewhere? Would they bind it at the same place as everything else binds it, or would they bind it at a different place? They have their choice of the entire surface. So the question is, will a random set of peptides bind at a random site, or will they end up binding at the exact same site? By the way, there will be quiz questions on this, okay? So I see that you're taking notes. That's good. The quiz questions will be written so that you can answer with some recall, but you will be wanting to look over your notes before the quiz. So that's really good stuff. I also am recording this, and I will post it just in case you want to go back and catch up on a particular part of it. Okay. So how will the interaction work? So what will the interaction be like if you look at the amino acids that are there? Is there a particular kind of amino acid that works well for binding a lot of different things? So this is the title of the paper. And the title, like all good titles, gives you a result. Convergent solutions to binding at a protein-protein interface. You made the random stuff and it converged onto the same binding site as all the biologically relevant binding events. So this is what it means by convergent. The random, a divergent group of random sequences nonetheless converged on binding the same site as everyone else. And so when you look at this, look at the author list. And more important than the author list, so the author list, there's two people I want to point out. Warren Delano, the first author, the one who did all the work. James A. Wells, the one who ran the laboratory and who did a lot of work as well, but at a higher level. The one who was actually in the lab versus the one who wrote the grant, okay? The last author is the one who wrote the grant. And you see back there was James A. Wells. There's a couple of clues you can get about the history of this, and I'm actually going to turn it over to you. Look at the institutions and look at even the email address, and there's some clues about the history of James A. Wells. What can you see that stands out as interesting? And I guess the one thing that I will point out, this could very well have a 1,2 there. So James A. Wells was actually at both places, but you can tell something about the sequence, the order in which he visited those places, if his email says this now when he was at both places. Any observations, anything simple or complex? Just okay, so where you see the yeah, and you notice that the pharmacy shows up in an industrial or an academic context. It's not really a department of pharmacy, right? It's a pharmaceutical company, so they're looking to make money, okay? So one of the questions you can ask yourself, why did this come out of an industrial context rather than an academic one? At least you have 2 industrial places and 1 academic, and you would be right to say that that's a high concentration of industrial type stuff for an academic question. You know, a counterfactual game does not make you money. So follow the money. Okay, Luke, what do you see? Okay. Right, right. And the other thing is, because it's an academic title with an industrial thing that's making money, actually if you look through it, it's not easy to find how this is making them money. When we get to the new paper, you'll see the long-term plan of James Wells, how it all works and fits together. But for this one, this is actually basic research. And this isn't making them much money. James Wells was famous because Genentech hired him on, and they gave him a lab, and they told him, figure out cool stuff about how proteins work. And then they let him alone. And he did stuff like this, which is a cool paper that I still admire today. So every academic was jealous of him, right? Because you don't have to fight for grants, and you don't have to make money either. Nonetheless, he left Genentech and went to Synesis, and now he's at a new company again. So that tells me that all of these, academic riches are fleeting. Even the best jealous situation, for whatever reason, he left it. So it must not have been that great, right? But you can tell a little bit about the history. So the high concentration of things, notice that Genentech here, he was at Genentech, then he moved to Synesis. Genentech's going to show up in the Emperor of All Maladies rather prominently. And so it's another reason why I want to tell you this story, and you'll see more about them as we go along. But of course, this is a long time ago. This does not have, I don't think the word cancer shows up in this, you know? So it's not obvious what the connections are. So let's make them. Let's make the connections. Any other observations, anything else that you want to see? I think that's most of what I saw, but you might see something else in it or have a question about it. Definitely for all of this is if you have a question about the things that are on the screen, I hope to be able to address it. The other thing is, here you see James Wells writing an obituary. They don't have many obituaries in science magazines, and especially not for first authors. You know, the first authors, when first authors pass away, it's tragic, and it's an early death. Warren Delano died early. But if you look at what he did, the first author, after doing this, he moved on to do something that you're already familiar with. He was a programmer. It was like playing a musical instrument. The programs he designed were symphonies in computational space. These masterpieces can be played by everyone. Well, that sounds like hyperbole. Can it really be played by everyone? This is the program that he wrote. So I think you can guarantee at least everyone in this room that you can play Delano's programming masterpiece because you already have. PyMol was what Delano did. And if you look at the startup screen, it's made by Delano Scientific. In fact, it's still called Delano Scientific even after his passing. So, and really, it's PyMol. We use it because everyone uses it. It's simple and easy to use, and it was early on free, and now it's still cheap, you know. And as a student, I believe you can still use it to your heart's content. So all that's to say, this is how Delano got his start before he decided to go ahead and commercialize PyMol. And you'll see some early PyMol figures if you look through the figures of this. So the part of the protein, the part of the antibody that they're focusing on is the FC region. Notice that the antibody has a Y shape. Antibody binds antigen at an epitope. An epitope is the surface of the antigen that the antibody grabs onto. An FAB is so an FAB is the Y part, the arms part of the Y, and the FC is the constant part. So this part has to stay the same so that immune cells, like FC gamma receptors, you can see this even though it's in German. By the way, this is in German. You don't even know until you look at the bottom, but that's because scientific language is relatively universal. There's a receptor on a granulocyte, which is a kind of immune cell, that grabs onto the FC region, and that is how a granulocyte is able to grab onto an antigen. Okay? This antibody is a protein. It's made of amino acids and a little carbohydrate. And it varies, it's able to bind any antigen because it varies its binding site. It varies which amino acids are able to bind. This is a protein binding problem. And so this is what they were looking at. Now, the thing is the FC receptor can bind to the constant region. The constant region is pretty big, though. You know, we need to look and see, okay, what all can bind to that region. You know, there are lots of places where things could possibly bind. Well, it turns out that bacteria have figured out how to bind the constant region because it's constant, and if they make a binding protein, they can mess with it. And if they mess with it, they mess with all the human antibodies. Okay, so there has been some convergent evolution among antibody binding. Immunoglobulin G is the type of antibody that is most common. Antibody binding bacterial proteins. And it turns out that different bacteria bind constant regions in the same way, even though they don't have to. That's why it's convergent evolution. Here's the abstract of this, and this is the kind of thing that you want to pull out of the abstract. Okay? Right here it starts with protein G is a bacterial protein that binds IgG FC regions. So it binds the FC region of antibodies. Okay? Moreover, so the protein G is a protein that binds antibodies. There are two other IgFC binding bacterial proteins, proteins A and H, which also bind FC regions. And they don't have any sequences that look the same. When you line up the sequences, they don't line up at all. And they also, each of these three proteins inhibited the other protein. If you throw in protein H, it'll block protein G. Protein A will block protein G, you know, likewise. And so what they did is they actually took this and they wanted to find out why do these three proteins with different sequences block each other in binding? Do we have three very different sequences on one side binding the same site at the other side? And it turns out that's exactly what we have. Okay. This is an unusually clear example of convergent evolution. And so that's another reason I'm interested in it. I think convergent evolution is pretty cool, and I want to know how it works. And here we have very different proteins that converge on their function. Their function is to bind the FC region, and more specifically to block the immune system binding to it possibly as well. So we have protein A, protein G, and protein H. They're all different proteins with different sequences. The Theodor Geisel protein naming convention, that's a little joke because Theodor Geisel, do you know who Theodor Geisel is? It's a doctor, Dr. Seuss. That was his real name. So it's like thing 1 and thing 2, protein A, protein G, protein H. And it just reminds me that we had this, they're too big for these now, but so I have to show the pictures. But when you have 4 children, then you have to eventually get them things like this so that you can name them after the Dr. Seuss characters. And now they have a shirt that you can get that's Mother of All Things in the same script. But now they've gotten too big, and we've had to give the shirts away to a family that has 3 kids. So there you go. But anyways, protein A, protein G, and protein H, at least it's not complex names. They're all bacterial. They all bind the Fc region of human IgG antibodies. None of them have any detectable homology, and all have totally different domain structures and protein folds. So they're different at every structural level, primary, secondary, and tertiary. And yet they have the same function, which is to bind the Fc region, and they block each other. So this is how they did it, and if you look at their abstract, they talk about the protein G is actually pretty big, but it's got a bunch of domains in it. You see here it's like 1 to 593. But it turns out that if you slice it up into its domains, the domains still function, and you can slice it down to just this domain, just this 60 residue amino acid domain, and it still folds, it still blocks binding, still binds the Fc. And you can predict what its secondary structure is. It's got 2 betas, an alpha, and 2 betas following it. You can predict where the turns are. So this is secondary structure prediction. And then you can even take it and make little peptides to see which of those peptides might be able to block its function. And so they made all these peptides of different lengths that correspond to these different amino acids and these different secondary structure elements. And they found that, these didn't block, still work. And then you throw these peptides in, or I'm saying it backwards. The plus means it blocks, or the plus means that this peptide binds the IgG. So you basically cut the sequence down. The whole thing binds. This shorter one binds. This shorter one binds. This shorter one binds. And so let's take all of them where they overlap, make a peptide just with that. And it turns out if you make other peptides from different sections, they don't bind. Bingo, it binds. So you've got a very short little length that's only about 12 residues long that still binds FCC. So you've really narrowed it down to what part actually does the binding. And you can see the sequence down there. What's more interesting is to map the part that binds, map the binding peptide onto the domain structure. This is relatively easy to solve. This is protein G, and it turns out that the part that binds is a loop between this beta strand and this alpha helix. Just this part of the protein will bind. So this clearly is the part that's fitting into the binding site somewhere on the Fc region. Okay? So we've really narrowed down the binding site in this case. And so one of the questions is, okay, protein G binds with a loop. Maybe the other ones bind with a loop as well. Maybe it's a loop held, maybe the loop has similar sequence. There's already a hint that this isn't true because the other sequences aren't anything like the sequence of this loop. So all these protein G, protein H, and protein A all bind, but it turns out that the secondary structure and the primary sequence of the binding peptide that actually fits into the binding site, totally different still. So we haven't gotten any nearer to understanding how this works. Why are bacteria able to bind at this particular place? So let's look at other things that bind the Fc regions of antibodies. So these staphylococcal proteins do. We've got 3, protein A, G, and H. Fc receptors on the surface of immune cells will bind the Fc region. We showed the granulocyte doing that. There's a neonatal Fc receptor in the fetus, which is how the fetus actually gets antibodies. It gets antibodies from its mother's milk by binding the antibodies in its stomach and moving them through to its bloodstream. There's actually a receptor for this in the gut that will do it. It's one of the reasons why infant stomachs are not acidic, because they don't want to blow up the antibodies. So there's a receptor that grabs onto antibodies, and of course it grabs onto the constant region because that's where you want to grab it. There's there's an immune cell receptor. The biggest one is called Fc gamma R3, and that's one of how immune cells grab onto it. There's another thing that binds it. Other antibodies sometimes accidentally bind the constant region. Now, if you have this, you have something wrong going on, because if you make an antibody to an antibody, especially if it's the constant region of the antibodies, you're going to be self-binding your own antibodies. And that's how you get rheumatoid arthritis. So rheumatoid factor is one way to get rheumatoid arthritis. It's an antibody-antibody interaction. And so this is interesting. Why does the other antibody bind the Fc region? Could it be because the Fc region has a particularly sticky spot that antibodies are prone to bind to, that anything's prone to bind to? And the question is, what would a completely new protein do? Just to let you know, all of these bind at the same site except this one. So one of these binds at a different site. The Fc gamma R3 receptor binds at a separate site on Fc region. But this, and this all bind at the same site. So you have 7 things, different sequences, same site. So let's take a look at it. We've already talked about protein G. It has that loop that we talked about. And when you have the protein G binding the Fc region, the Fc region is actually a dimer of these beta sheet regions. And so C is for the constant part of it. So the Fc region is Fc, and we'll talk more about what the H means on Friday. But you have the 2 domains, and the protein G region binds right at the knee between the 2 domains. And you see there's the loop sort of fitting in the way it does. The green residues are the contacts, where the domain here contacts the domain there. Protein A uses an alpha helix, so a different secondary structure, different primary structure, and its binding site is shifted over a little bit. It's binding some of the same things, but they don't really overlap. It doesn't even contact necessarily all the same residues. So you've got protein G and protein A, the binding sites overlap, everything over here overlaps, nothing out here overlaps. So you can't say that they're both using a protein G-like loop in the same way. There's no secondary structure similarities on the binding protein. You can also look at the primary level, no sequence homology. Basically, I can't show you a sequence comparison because there is none that you can find. Maybe they're the same kind of interactions. Maybe they're both charged interfaces. Well, you look at this one, there's a lot of charged residues. Okay, maybe we're getting somewhere. You look at this one, it's a hydrophobic interface. So they're totally different kinds of residues. And so, you can't make that argument. The only thing that you can exclude from our list previously, by the way, is the Fc gamma R3. And it actually binds the middle of the antibody. So you see the other ones were binding down here, sort of at the knee between CH3 and CH4. This is actually binding, actually the knee between CH2 and CH3. This is binding sort of in the middle. The rest of the antibody goes up like this. And so Fc gamma receptors are different. There's something about, and that may be constrained by biology. Maybe something about the way the immune system works, that it needs to grab the middle of the antibody. Everything else, we have 6 or 7 different examples that they all target this part right there. Why is that? And possibly part of the reason that the Fc gamma 3 binds at this point is it leaves the Fc region, the whole Fc region open, leaves that site open for other things to bind. In fact, that's probably the reason why it does that. So we can't even say that that is really an exception to the rule, because it looks like it's leaving the sticky site open for other people to bind. In fact, they have some mechanism about how that might work. They have other receptors that might bind. This is all immunology, and we won't talk about that too much today. But let's look at the proteins. So let's compare 4 of these different binding sites. Here's protein A and protein G again, and really lined up this time. So here's protein G, there's the loop, there's protein A, it binds with the helix. Here is rheumatoid factor, and you notice all the beta sheet domains? This is what an antibody looks like, okay? This is an antibody binding domain looks like, and it's an antibody binding domain that binds that same site. So rheumatoid factor binds that same site, even though it's not supposed to. It causes a disease, right? And then you have the neonatal Fc receptor, which binds the same site. So it looks like the Fc gamma receptor is the odd one out. And in all these cases, you look at it and you try to figure out, Is there a simple rule for why they keep binding at that site? You can't find a simple rule. And that's why they wanted to let's do a random peptide. In a sense, the bacteria have already done random peptides, in a sense, because they've tried different things and they've found that these things stick through convergent evolution. That is, that's actually the neonatal Fc receptor. A little tidbit about it. It is actually a MHC-like protein. For people who've been through immunology, you talk about MHC proteins in a totally different branch of the immune system. And yet somehow the fold has been repurposed to act as this weird neonatal Fc receptor that gets antibodies across the infant gut. Really cool example of how you can use a fold for a very different function. But that's neither here nor there. I want to talk about how they actually did this study. Okay. So you look at the study, and you might be able to find this in your paper itself. They performed an in vitro selection, so in test tubes, isolate peptides that bound Fc without the constraint that the peptides function in vivo. We're just looking for binding. There's no survival constraint, no biological mandate. They made cyclic peptides consisting of 4 × 10⁹ different peptides of the form XIC, XJC, XK, where C is a cysteine, so that's why they made them cyclic. They made cysteines that bound each other, made a little loop. Give it a little bit of structure so it would have some sturdiness to be able to bind. And then they had XB random amino acids, and they just had a constraint that I plus J plus K had to add up to be 18, so they were all 20 residues long. But the C's could be in different places. Realize that I could be big or small. So what they say is they made a library of different peptides, and they expressed them on the surface of M13 bacteriophage. Why are we talking about phages all of a sudden? They're NH2 terminal fusions to the gene 8 protein, and they're selected for binding to the immobilized Fc. So what does this mean? The gene 8 protein is part of the bacteriophage, and the bacteriophage is what kind of organism? What branch of life does a bacteriophage come from? If you just take apart the name, it doesn't help too much, because bacteriophage just means bacteria eater, and that's actually not a very accurate description. Any idea what a bacteriophage is? Exactly. And so you're using the fact that it's a virus because viruses are just protein. They're relatively simple. They're just protein and a nucleic acid code. And so you're actually using the virus infection. So you're deliberately infecting bacteria in a sense here. You can do this with a large population of bacteria and a large population of viruses. And it's actually pretty cool. So you had all these ones, and you were able to select the ones that bound immobilized Fc. You had a surface with Fc on it, and some of these random peptides stuck to it. When they stuck to it, they were bacteriophages. So they were stuck to it. They were on a coat protein. There's a coat of protein around a nucleic acid core. The nucleic acid core contains the information to make the protein. So that's the cool thing about this virus. You have a DNA tag because it's on the surface of the coat protein of the virus, has the random protein. When it sticks, it has a DNA code that's attached to it. You purify that out, and you can do PCR that up. You can do all the DNA stuff to it. The DNA works, the DNA of the virus works as a tag. If you look at this a little bit closer, you can see how they actually did this. They made J equal 4 to 10. They give you a little more detail for the random thing here. And then they had a glycine-serine linker, and then they had the M13 gene. So what we need to do, if I was facing this and I really didn't know what was going on with bacteriophage selection, I would look up phage display, or how phages work. That's the name for the technique. Bacteriophage is used exactly because it infects bacteria. When it infects bacteria, it makes more phages. And so that gives you an automatic amplification for this, and you can actually do a cycle like this. You could take a library on the phage. Right here it talks about how you can put an antibody on it. Here they just put a random 20 residue protein on it. And then they panned it over a column, an affinity column, with the FC region on it. The red ones stuck, the blue ones didn't stick. The ones that stick, you elute it, then you amplify it, which means you just grow more of those phages. You make a new library, and then you pan the cycle again. Each time you run it through the column, you enrich it for the binding. You know, some of the binding might be strong, some might be weak, but as you do this more and more times, you enrich, you get more and more of the red proteins. The tighter binders you'll get more of. And so eventually you can just take this off and you can sequence the DNA in the phage, and from the DNA sequence that stuck, you can tell what the protein was. And that's what they did. This is going to show up in Wells's other thing. This is one of his hallmarks of what he does. So the cool thing is that 2 different phage display experiments produce convergent results. So they did this in principle, and they say that, you know, they didn't limit it to a particular region of the FC. Peptides could have gone anywhere on the FC that they wanted. The library was random and unbiased. Several rounds of selection, the library became dominated by a single peptide. So they did that over and over again, and they sequenced the DNA, and they got a single peptide each time. They did it once, and they got this peptide. They get it once, and they got that peptide. 2 independent experiments. So what do you notice about look for the cysteines. The cysteines are in about the same place. They're about 5 in from the end, each case, even though that could have varied. And look, do you see the sequence similarity in the middle? The GLVW sequence is the same in the middle. So it looks like there's something about, from the random peptides, there's something about this sequence that binds especially tightly. There's been no restriction put on for where it binds the FC. So then they checked that they got real results because they did the experiment in a different way. They made peptides. You can synthesize peptides with FMOC chemistry. So they did that, and they found that if they threw in the peptides, that the protein A would bind FC less. And they did an inhibition constant, which is like a dissociation constant. It's like a KD. And they found that it was about 5 micromolar, so low micromolar affinity. That implies that these peptides bind to an overlapping site. So it implies that they bind to the same site that the protein A binds to. And remember, protein A, protein G, and protein H all bind the same site. So it sounds like we're converging on the same site. By the way, if you look in the methods for this, there you see FMOC chemistry. And so if you want to remember things about that, that's the part of biochemistry text that you go back to. They use reverse phase chromatography to purify the peptides, and they even confirmed the masses with electrospray mass spec. I did all this as a graduate student. If you have questions, ask me. So, but that's how you go through. The methods are still important to understanding and presenting your method. So you are actually going to be making your peptide. All right. So right here, they used ELISA, and actually I moved this around. Did you talk about ELISA? You've talked about it maybe in other classes. We will talk about it again on Friday. ELISA is a way to measure binding with antibodies. Okay, so that's going to come on Friday. I actually am doing this out of order, so I apologize. This is not the return of ELISA, but this is the prediction of future ELISA will come through. If you look at this, they immobilize it on microtiter plates, and we're going to talk about how ELISA works and all that. But the main thing is they did a mixture of the peptide. They mixed the peptide in with FC. They incubated it for an hour. Then they washed it, and they found out that how much peptide was still stuck. And they developed inhibition curves for that. And they ended up, that's how they obtained the Ki. And so you notice that the Ki requires concentrations of peptide that are around the affinity. See, 5 micromolar affinity requires up and down above 5 micromolar of the peptide. That's how much you need to measure the binding. We'll talk more about exactly what the microtiter wells are doing here. This is using an antibody. You see right there, actually, I'll talk more about what ELISA is. They used ELISA to measure the Ki. So they didn't stop there, though. So that peptide was good. But they actually wanted to do another few rounds of affinity because 5 micromolar is actually still pretty weak. Okay? They wanted to see if they could get down to nanomolar, and they could because they did the gene display a few more times. So basically they took the FC2 sequence, the second of the 2 identical looking sequences, and they did another round of phage display where they randomly mutated 5 residue blocks to exhaustively cover all the non-cysteine positions. In other words, 4 times 10 to the 9th sounds like a lot of sequences, and it was random, but it wasn't a complete coverage. If you want to figure out how much sequence space you could have covered, then you should take 18 or 20 to the 18th is the number of sequences, possible sequences you can have for an 18 residue peptide. That's a lot lower than 4 times 10 to the 9th. And so they went to, but you can be exhaustive if you do it in smaller blocks. And so that's what they did, 5 residue blocks. They did 6 separate libraries. And they basically took the residues that came out of those, they recombined those, they gave 3 more libraries spanning the peptide sequence. And finally, they got down to a 13 residue core that they kept getting out of these. And look again, you have this part on the side which looks different, GELVW. That's the same sequence that originally showed up from FC1 and FC2. They call this FC3. So this is the 3rd result. They wanted to see how tightly it bound, and it's tighter binding. 25 nanomolar binding. So 25 nanomolar concentration of this will block the binding of protein A. That's better than the 5 micromolar concentration that did. So even though it's 7 residues shorter, it binds 200 times more tightly, 5 micromolar to 25 nanomolar. Despite its smaller size, if you compare the affinity, it's only about 2-fold weaker, only slightly weaker than the affinity of protein A and protein G domains, which are each about 4 times larger, and they bind with KDs around 10 nanomolar. So in other words, the bacterially evolved proteins that the bacteria depend on to bind FC, 10 nanomolar. With this random thing, they got down to 25 nanomolar. And if you look at the math, the fact that it's smaller also means that, that they bind pretty much about the same, because size matters for these calculations. Okay. So this is where they called it a day. Okay, they said that we've got the peptide, now we want to look at it. They did a protein crystallography study. They crystallized it. And I just want you to look at this again. Actually, let me ask you. We talked briefly about how to interpret these before. Do you remember what the one biggest number out of all this that you should look at is? Exactly. And so, and it's the smallest resolution number. So you see right there is 2.7. Interpret that. Is that a super good resolution at which we will be able to see like every atom and every hydrogen bond? Kind of a leading question, but it's late in the day, so I'm going to be a little leading. It's also not super terrible. You can see where most of the big atoms are. You probably can't be certain of where the residues are rotated, and therefore you can't be certain of hydrogen bonds. What do you see? Yeah. Yeah. Yeah, it typically is about 2 angstroms, but it's not so much the length that's an issue. Covalent bonds, they have to follow the hybridization rules and everything, and they have to be connected. So you are pretty certain what the angles and all that are. The single bond angles are the things you aren't certain of. And so you can be certain, and you can do the primary sequence, and you can get enough information of that you can follow the primary sequence. And you can tell that I've got an alanine here, I have a tryptophan there. Those are big enough. But you can't tell. Hydrogen bonds depend on hydrogens, which you can't see at 2.7 angstroms. And they depend on the orientation of a nitrogen and an oxygen, for example. And if you can't really tell which way these 2 atoms are rotated, you can't tell whether you have them pointed straight at each other, forming a hydrogen bond, or if they don't care and they're like pointing like this. You just don't have the resolution to do that. Yeah. But you can tell what's in the right place. You just can't tell. You got to remember that they can move. And it turns out that that's important for how this works. So boil this down to the 2.7 number. You also can look at the R's, and the lower the R, the better. Although in general, these are around 20 to 10% is good. And just to say this, you see that these R values are around 20%, so it's on the high end, but it's also not terrible, and it probably was hard to crystallize, and they just have to be a little careful. They can't talk about hydrogen bonds that much. Okay. But they can see where it binds. And consistent with the inhibition experiments, it binds right there like everything else. So now they want to say, okay, it looks like it really is, this site is sticky. And yet it's sticky to things that are very chemically different. Why does this random peptide bind so tightly at the same site as everyone else? It's not peer pressure. It's something inherent in the antibody that makes this site a good binding site. So you can see right here it says the FC3 peptide covers almost as much area on the surface of FC as do the fourfold larger FC binding domains. So it's actually, even though it's small, it binds at the same site and it covers about as much area as the protein A and protein G do. So there it is at the same site, covering about the same area. We want to take a closer look at the comparison of the different areas, though, and so that's what they do. There it is. So you look at it, and there's a cycle. Look for the cysteines. Can you find the cysteines? Looks like they have them this weird sort of red color. You never know what color cysteines are going to be because they can be all sorts of different colors. There they are, forming the covalent link, making it a cyclic peptide. And if you look carefully, remember that it's got the GELVW sequence. Can you find the GELVW? It's actually a little bit tricky to see here. You can find the W's, right? There's one here, and there's one here. And it turns out if you look, GELVW, I believe the GELVW's on this side. I think here's the L. And then the V, you can't really see. There's the V sort of sticking down. These are so complex. There's always a lot going on. And I think the VW is here. So this is the GELVW. And it just fits into a nice little groove right there. And they say right here, our conclusion from looking at the numbers was right. The resolution of the structure is not sufficient to conclusively identify hydrogen bonding interactions. However, we're going to go ahead and say it anyways, because we're pretty sure. It appears that 8 hydrogen bonds are probably formed between the peptide and FC, including those involved in salt bridges, because salt bridges can also be hydrogen bonds. And so they think they found 8 hydrogen bonds, which comes close. That's a lot of hydrogen bonds for just 20 residues. So that comes close to saying how this binds. It uses hydrogen bonds. Very directional, but they can also be formed by residues rotating. So let's look at the surface areas. And what they did for this is they did 2 graphs. This is a graph. Remember that we have protein A, protein G, and rheumatoid factor. We have crystal structures of those binding to the same site. They actually took the FC, the neonatal FC receptor was a 4th example that they had on the previous slide. But look at its resolution, 4.5 angstroms. So they just said, we don't need that one. They threw it out. And they basically said for these previously solved structures, where all 3 of them touch the protein, let's color that red. Where 2 of them touch the protein, we'll color that yellow. And where just 1 of them touches the protein, we'll color that blue. And you see it makes sort of a target centered on this groove right here. So this is the binding site right here, where everyone loves to bind the protein. And if you look at where the peptide, the FC3 peptide binds, and you color its binding surface green, it's like it's a target that it just hits. It's like, and I think they deliberately chose these colors to make it look like an archery target or something like that, because it looks like it hits the bullseye. You can't see much red or yellow peeking out, and there's just a little bit of blue. Like this is the binding part, And this is where things like to bind. So why do they like to bind there? And why do both hydrophobic and charged things like to bind in the same place? This is where they go and they look at the actual groove and they look at the residues lining the groove. Okay? Hydrogen bonding sites are shown with diagonal lines, and salt bridge locations are denoted by open circles. And so you look up and down. Well, make an observation. Compare here's the bottom of the groove. Here's the sides of the groove. And this whole hatch is where the peptide binds. So it sits down on the groove, but it also covers the edge of the groove. Make observations about the kind of residues that are on the edge versus the ones that are in the middle of the groove that are important for binding. What do you see? Yeah, so you see the, you see the histidine, and that's polar. It's not fully charged, right? Methionine, which is kind of a weird category, it's sulfur, and it's polarizable, but it's not really, it's like on the polar end of hydrophobic. So you have something on the hydrophobic end of polar, and you have something on the polar end of hydrophobic. And those are right in the middle of the target. And then did you see something, what do you see on the outsides here? This is a different type of residues. Yeah, these are the small hydrophobic. It's unusual to see those sort of sticking out of a protein. So it's kind of like you're right on the edges of all your categories. You have kind of small hydrophobics on here. Tyrosine can be hydrophobic, and it also has that little, but you look here, it's presenting a hydrophobic surface. So you kind of have a hydrophobic rim. You kind of have three areas. You have the interior, which is the methionine, which is hydrophobic, but it's sulfur. So it's kind of, it's not like carbon hydrophobic, it's sulfur hydrophobic. So it looks like there's methionines at the middle. There's some exposed hydrophobics, but not too much. And then you see out here on the edges. Look at the edges. What do you observe about these residues out here? You see charges all the way on the outside. And the charges are sort of sticking up. Remember that we said residues can rotate and they can form hydrogen bonds? We don't know for sure, but it sure looks like they're able to form a lot of different hydrogen bonds. So it looks like we have a special situation where we're using special residues in unique ways, and there's sort of a three-layer structure to this. There's a groove with the methionine and histidine in there, and then you have the hydrophobic middle of it, and then on the far outside you have these hydrogen bonding and fully charged residues which can form sort of exterior. So just like you had a target with three different colors that they showed earlier, there's kind of like a target with three different chemistries that they show here. Now this is the thing I can't show you. I tried to work this up, and somehow this is such an old .mov file that nothing can play it anymore. But what they do is they show you the groove for the peptide here, and then they rotate it around and they change it to the contact residues for the three other proteins that they're comparing, protein A, protein G, and rheumatoid factor. And the thing that you'll notice is that the side chains are very different. Of the yellow stick side chains, the stuff that's binding to it is very, very different. There's no similarities you can see along there. But of course the groove is the same. But if you look up and down, I just want you to look up and down and notice that, like the methionines and histidine, the bottom of the groove doesn't change much. The outside of the groove can change a little bit. It like molds a little bit, like this is the hydrophobic part out there. And then out here at the edge, you have the charged stuff at the edge that's still forming good interactions. And those are rotating all over the place. So you kind of have this three-part structure with the middle that can't move at all, the hydrophobic rim that can move a little bit, and then you have these adjustable charges on the outside that can really move like crazy and bind stuff. Okay? You can really see that well in the movie, and if I can find it, this website does not work anymore. It gives you a 404 error. So, I saved it because I was worried that would happen, and now the movie file is too old. I'll try to pull it up, see if I can find an alternate version. I guess we've just gotten too old with our science now. So that's the movie. The movie is like this. It rotates. But the main thing you'll notice as the movie goes around, as you go to different ligands, these residues rotate a lot, these residues rotate a little, and of course the stuff in the middle doesn't rotate at all. And that's how you can do it. You can have the charged residues that are important in some cases, and the groove that's important in other cases. It just looks like that's super bindable. But they wanted to come up with a rule. We're scientists. We want to have rules, right? And so they wanted to come up with a rule for how can we predict if a patch of protein, if a surface patch will be bindable like this patch is bindable. And so they wanted to come up with a way you could put it into a computer. This is what they came up with. They took all the patches of the protein and they calculated their accessibility. They took all the patches of the protein, they calculated their polarity. And they asked, out of all the patches of the protein, where does our one important patch, our binding patch, where does it fall? On accessibility, it's not the most accessible, but you see it's definitely a standard deviation above average accessibility. So it's more accessible than average, and it's less polar than average, okay? Put those two things together. Do you expect those two things to go together normally? Do you expect most accessible patches to be polar or non-polar? If you have something that's accessible, that means it's accessible to water, right? And so what would you expect for the polarity of a patch that's accessible to water? Yeah, so you'd expect the same side of this, but instead they're on opposite sides. So we have sort of a paradox. And this is, I like that they found this because this is kind of it shows why this site is special. So also notice what's not there. Actually what's not there is even though they detected 8 potential hydrogen bonds to the peptide, most of the surface is not capable of hydrogen bonding. This goes with its low polarity, by the way. Polarity makes hydrogen bonds. And so with this observation goes with the fact that only 19% of the surface was capable of hydrogen bonding compared with 37% on average. So that means that you don't need too many polar interactions. Actually having that hydrophobic rim was probably a good thing for this site. It's just it's energetically difficult to have a site like that on a protein. Maybe antibodies evolved to be super binders at that site. It's possible. And they actually took all the patches, they tried to color it by red, all the parts of the protein that had high accessibility and low polarity. And so they colored it red, and it turns out that our special site lights up red. These other things light up red, but none of them are a groove that has the geometry that allows something to really bind in there like this. The other site that shows up red, I find this interesting, is the place where the Fc gamma R3 receptor binds. Remember that it binds at a separate site? It also binds at a red lit up site. Now, I'm not sure how much to make of it because they don't say anything about it, but that's the way it is. Methionine is important. We've already talked about that, and the methionines can move a little bit, and there's 2 of them right next to each other. Those are relatively rare, and considering that they're sitting in the bullseye, don't forget that the center of the bullseye is probably important as well. And finally, this is probably something that a reviewer asked for. Reviewers always ask for some kind of question to make some kind of experiment, which is particularly frustrating when you're at an undergraduate institution, you don't have graduate students working for you, and they ask you to do a whole nother experiment to be able to accept the paper, and you're like, I've got to find a new undergrad, to do this new experiment. Can't just give it to the same person because they're already graduated, so it's that kind of thing. But they did a thing where they actually made peptides. Remember, it's peptides, they're easy to make, and they replaced all the residues with alanine, and they found that the big residues on the peptide, if you replace them with alanine, it doesn't bind anymore. And they express that with a delta G of binding. And those are big delta G numbers. Those are big changes when you put the alanine on. And so this is how they really nailed it down that this is what's going on. This smells exactly like a reviewer question. Maybe you'll be able to identify one of those in your paper. So the thing is, the way this works, this is not exactly a protein design paper, but it does talk about how the site walks a fine line between accessibility and hydrophobicity. It is both very accessible and somewhat hydrophobic at the same time, somewhat non-polar at the very least. And so it's kind of like walking between too inaccessible and too hydrophobic would be less ideal binding sites. It makes itself an ideal binding site by walking the edge between those two things, and by using amino acids that are located on the edge of our chemical categories. So I think that's really cool. Okay. And one extension I can make, feel free to make these kind of extensions. I will not mark off for wild speculation as long as it's good wild speculation. Could we use this peptide as a therapy against rheumatoid factor? If you put it in, could it stop the antibody from binding its own antibodies? I think it's possible. Peptide binds the Fc of antibodies. Maybe it could block the site from the antibody binding. The problem is peptides are awful therapeutic agents. Can you give me a reason why a peptide would be much worse than, say, a small drug like aspirin for a therapy? Yeah, the flexibility of it. I mean, you can tie it down a little bit with the cysteine, but still, and the big thing, denature really easily. Also, hydrolyze really easily. You have proteases that go around trying to chew up every protein they can. Yeah. And also, if you think about it, this could possibly interfere with the neonatal Fc receptor. So it would have to be one of those drugs that you would not give to nursing mothers. Okay. There's another story. I never have time for this anymore. But they did a randomized protein that bound protein A very well. So they randomized the other side of things. And guess what? It bound protein A at the same binding site. So it looks like protein A has a favorite side, you know, a good side that it presents for binding. Okay? This is all about, the random proteins are called affibodies. There's a bunch of papers about it. Actually, you might find an affibody paper in your protein design. And if you're still looking for a protein design paper, consider looking for affibodies. They try to make alpha helical random peptides that work as antibodies. But I want to skip over this side as well. But this is what James Wells did. Now he's still at UCSF, at least. He's settled down a little bit. And if you look at his research summary, he spent 16 years at Genentech and then 8 years at Sunesis. And that's the company he founded. Maybe that's why he left Genentech, to start his own stuff. But then he went to UCSF for good. At Genentech, this is what he did. At Sunesis, they did drug discovery. And now he's working at all this stuff. And if you look at the very bottom of this, where is it? Yeah. He also built the Antibiome Center for Generation of Renewable Recombinant Antibodies. That is the connection to our next talk. So, to get ready for this, I'm going to have another paper. This one's 26 pages long, so I'm not going to print it out, and I'm not going to require that you print it out. You might want to print out certain pages of it. I don't know. Look over this paper. We'll discuss how to read it. There's phage display in it as well. It's posted on Canvas, and he uses an antibot, and we'll talk about how he uses the antibot in the next lecture. Okay? Your other homework is to hear him in his own words. He talks about drug discovery, and I think this is really good for you to, because we talk about biochemistry a lot, disconnected from industry. This is about him and a co-presenter talking about the drug discovery process, and this is what you can actually get a job in. And one of my goals is for you, if you're interviewing about a position and they talk about some drug discovery stuff, I don't want you to say, I've never heard that before. Here you can hear it from James Wells. And so this is about an hour to watch this together. This is about small molecule design, but you may encounter some of the ideas in protein design as well. You don't need to read this paper in detail, but I want you to be familiar with it, look over the figures. We will talk about it the same way I talked about this paper on Friday. Okay? Yeah. Any questions? All right, so check that out. And I will see you on Friday. This is sort of part one. The quiz probably will include a question or two on today, a question or two on the talk, and a question or two on the next one as well. And you'll be able to choose from those. So that's the level at which you need to know these things. All right, let's hope that recorded. And let's see.